So biotechnology has been in use since 1919. So it's a long time back, and it has its first application was in DNA recombination experiment with bacteria, right? And today, biotechnology in itself has been applied in manufacturing of medicines, medicinal devices, diagnostic tools, biomaterials, food technology, and food production, all of that. But today we wanted to talk to you about another exciting field, which is computational biology. So computational biology, which is a relatively newer field of research, has been evolving at an exponential pace. And it's such an exciting field of biology. And we will learn more about it in today's session. Uh, and to talk a little bit about computational biology, there's a reason that this exponential pace that it has got. It is because of the enormous amount of biological data that has accumulated over the years, and the size of this data is ever growing, and the application of biology in life in itself is growing already, and it has been shown even for the experiment that some scientists did a few months ago, that DNA can be used to store data just like our typical hard drives, right? Scientists were able to show that 700 terabytes of data could be stored in a single gram of DNA. So that's like pretty exciting, right? So, but our objective here is to simplify complex bioinformatics methods that have been applied in these amazing experiments. And then we want to use these methods effectively and efficiently to tackle unsolved problems in biology and in healthcare. So I would also like to tell you about in much more detail about the OmicsLogic program. But before I do that, I would like to ask if any one of you would like to have a discussion, a question, please feel free to put it in the chat option so that we can go through this later and answer your questions. So going back to the presentation and about the OmicsLogic programs and what we've been doing and been talking about. So the goal of OmicsLogic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer examples. Another aspect of important aspect of OmicsLogic program is to develop a growing community with a shared passion for data-driven research and application of citizen scientists science projects. So we have a growing community of over 10,000 people who are computational biologists, researchers, biologists, clinicians who interact with each other and enrich this information sharing and collaboration. The third objective is to enable students and clinicians and faculty of all background, people who are interested genuinely would want to learn more about it, apply these skill sets in their research areas for job opportunities, and to develop noble and independent research using life science or the latest technologies in bioinformatics and computational biology. So these are some of the objectives that we wanted to tell you all about. And the next thing is about the program itself, because we would want to complete some of these objectives by going through a training method. So this online program is designed for everyone, including students who don't have a background in bioinformatics. So the topics covered in this program enables anyone to learn the NGS data analysis, because we will be going through different kind of processing sections but for doing different sort of analysis, such as genomic analysis. And I've heard many of you are interested in transcriptomic analysis and interpreting the omics data using machine learning techniques. So we will learn about along with this, we will also be learning about public repositories where various file formats are being deposited by different laboratories and companies, and their application, and then understanding their application for biological research. So this analysis will include, we will be doing annotation, visualization, and interpretation of the results. So before we go into those technology, let me start today by mentioning that as a company in this space, we are focused on addressing some of the pressing needs in biotechnology, and in clinical market by providing user-friendly tools for researchers, clinicians, students to leverage this transformational opportunity of our lifetimes, because this is where the innovation is coming. This is where the latest technology is heading towards. Today, we will be learning about the overview of topics as promised that will be covered in the program. So let's start discussing about the basics of bioinformatics, big data, and these concepts and applications. So in bioinformatics, if we talk about big data, so the biological experiments produce multitude of data today. So compared to 20 years ago, so before that, we were using single experiments to generate data. Now there are thousands of experiments, automated experiments that are generating this multitude of data. And then we have open source databases like NCBI. Scientists from different part of the world can share their data and can collaborate with ease. So that's the new world. And we have now crossed the Excel barrier as well. So earlier we were used to use, I mean, most of the time we even use Excel, MS Excel for collaborating and sharing data. So bioinformatics researchers who are now are adapt with statistics, data, and programming on top of domain knowledge. There are also demands such as learning tools such as Panda, NumPy, Matplot, RShiny, all of that actually efficiently help us to maybe use machine learning as well. So this is right now is the market in bioinformatics. And there's a huge opportunity associated with it also, because it's a growing market, and it has a lot of demand for all the biomedical research that is ongoing, disease related research, and many more applications that today we will discuss. So learning about this, I mean, what is the gap, where we are, I mean, what is lacking and what we can do. So breakthroughs in molecular biology, advances of precision medicine, biotechnology, and agriculture have translated into advanced technologies, right? Because now we are generating data with ease, and that simplified generation of accurate and in-depth data is, is something like a blessing in a way. But at the same time, it poses like a lot of questions, because there's a lack of analytical power and appropriate skills that creates like a huge gap, because we have high quality data now, but we need experts to understand that data and make sense of that data. So those expertise and skill sets are lacking, and many of because of this, there's this gap between data and information in this field of computational biology and bioinformatics. And from a standpoint of what has been seen as, so bioinformaticians as such were viewed more like computer scientists before, who could manage a database, maybe run some algorithms. But modern bioinformaticians are different, and I'm going to tell you why. Because modern bioinformaticians have to identify what questions a researcher should ask, how to look for right answers. That's a very important part. And then what to do with this large amount of data produced from these studies. That is also a job of a bioinformatician to learn and understand. And also for all those people who are in biology and are now progressing towards learning more about biology and its application. So in bioinformatics now, as I was telling you about the opportunities, these scientists who are being able to do all of this, who can ask the research question, find data set, understand what answer that data set can give, these scientists are now integrated into every research team, making them into the core of innovation. So it's very important. So biology and as a field has been now ushering with the age of AI, big data with it, and creating a new line of scientists. That's where I'm coming at, which is bioinformaticians and computational biologists. So now about bioinformatics and the different kind of challenges. So when we talk about technical demand that prevents us from learning these skill sets because of the computational power that is sometimes required for us to do all this analysis. So in most of the cases, that means that our ability to analyze data and extract meaningful information is lagging behind because of the pace at which the new data is being generated. So oftentimes, like clinician, biologists, students from life science background report about challenges with data analysis and interpretation that is holding them back and limiting their ability to rely on advanced technologies such as genomic sequencing, which we could incorporate in our studies, in our research, and that could actually give us a really bright, clear picture of what is going on. So if I talk about bioinformatics and computational biology, the first three words that Wikipedia used to describe bioinformatics are it is an interdisciplinary field. So when we think about these three broad fields, for example, biology, statistics, computer science, or you can say instead of statistics, data science. But what exactly these disciplines are, and are there other fields within this disciplines? So when it comes to application in the context of biology, the current realm of molecular biology, the current realm of biology includes molecular biology, genetics, genomics, proteomics, metabolomics, systematic evolution, pharmacology, biomedicine, health sciences, and the list goes on. So this is not an exhaustive list, but when it comes to computer science, there are like obvious ones like software engineering, database management, information engineering, natural language processing, image processing. So these are some of the fields that are associated with the interdisciplinary nature of bioinformatics or computational biology, and how we can now being able to utilize these different areas of these different technologies into one area of biology. So what is bioinformatics? I think it's a convergence of biology and data where both are equally important. So we've been talking about data. So let's understand some of the data types, which are like some of the genetic data types, omics data types. So omics data includes phenomics, genomics, epigenomics, epigenomics technologies, and that can be used to. These technologies are so important in today's world because they can be used to explore the roles, relationships, actions of various types of molecules that makes up our cell, that makes up a cell of an organism. So many of this omics data can be generated using the NGS technology, which is commonly also known as high throughput sequencing. And this data shows, as I was telling you before, this data shows detailed information about genomic variants, pattern of gene isoform expression, and for an entire organism and for that entire time point. So the application of these technologies is something that is really exciting, and that we will be learning about in the next few slides. So one particularly exciting area of bioinformatics is its use in precision and personalized medicine. I think many of you have heard about it. So what is happening is doctors are learning more and more about the influences of the gene on a person's physiology, a person's metabolism, and more importantly, how drugs interact with their exact phenotypes. So these are some of the things. Other than that, using patients' personal genetic makeup and comparing it with the database of thousands of other patients, doctors can make more informed decisions about treatment and therapeutic choices. So these are some of the possibilities that are already happening in many places. Some of the drugs may be more efficient for a patient than others, while some of the drugs may have more unwanted side effects for some other patients. All these could be, theoretically can be evaluated now, given the hospital have maybe hospital or the organization have an access to those databases, and then they have the expertise to compare and find similar genotypes that could be treated with similar drugs. So there are different applications in medical sciences that could help make the entire healthcare system or the medical science more robust, empirical, and casual than correlational and uncertain, right? So most of such data types are generated randomly at a high speed. These sort of data sets are, generated at a really high speed, and these require like complex computational data processing system, a procedure to generate structured data sets which can be visualized and analyzed statistically. So that is some of the challenges that are associated with such high throughput data. But I mean, this is a program where we would want to simplify that and take you through this entire process of how we can actually do this by learning the logic. Now, this about this training program. So in this first training program, we will have live sessions every week. And the first live session will be about orientation and introduction to OmicsLogic coursework and access to the multi-omics user-friendly Bioinfo platform. So after the program registration, and many of you might have already registered for the program, the first week, we will have participants and we will have orientation and introduction session. In this session, we will be collaboratively working with students to complete their academic profile on Learn OmicsLogic and set up accounts on Big Data Analytical Bioinfo platform. So about this, I mean, anyone can join the OmicsLogic Bioinformatics community for free, and that we will be showing you in a while. So in this week, we will also learn about big data, application of bioinformatics, and after we do the sign up, which we will be doing in the next few slides, I'll be taking you through the hands-on courses and the premium courses associated with each week for you to learn all of this on your own. So in the next session, after the first orientation session and the introduction session, where we get to learn about you, about your interest, about the area that you would want to work on, maybe, if not about the basic training, the help that you need to go to the next level. So in the next session, we will be learning about this various NGS technologies, starting with genomics. We will learn to analyze NGS genomic data. And to give you an overview, genomics is an essential subfield of bioinformatics and is a major force in expanding human knowledge for genetically associated associations with disease and other traits. It's an interdisciplinary field again, and it includes development of methods for DNA sequencing as well as big data analysis for genomic sequences. So NGS data or NGS techniques allow whole genome sequencing and then analysis of epigenetic factors as epigenomics for DNA protein interaction, DNA methylation with, I mean, it is very efficient, and it has very efficient way to identify those. And there are massive databases. Obviously, I talked about NCBI, there's GeneBank, EMBL, SRA for you to do data-driven discovery and knowledge discovery. So this session serves as an introduction to the subdisciplinary field of genomics. Participants will get familiarized with biology of genetics, genetic variation, and other practical applications. And to tell you, I mean, I will be telling you more about this entire week once you all are signed up, so that we can have a demonstration of what will be covered. So in the next session, after learning about genomics, we will learn about metagenomics, or you will have an opportunity to learn about metagenomics. So this is a 1-month program, and I would want to clarify that you will have a lot of resources that you can complete in that period, or you can extend keep on extending and keep on learning about different data types. So yeah, obviously there's an opportunity to do this right now in a month's time, but I mean, many of you would want to go and deep dive and can do these analysis after subscribing for the program every month. So in this session, we will learn about application of microbiome metagenomic analysis in life sciences. And as you know, metagenomics is to study a collection of genetic materials or genomes from a mixed community of organisms. Metagenomics is usually refers to study of microbial community. And we have also different projects in metagenomics to explain different sort of analysis that I'll take you through after the login. So after the metagenomics section, we will move to another section of analysis, which is transcriptomics analysis. This is like high throughput gene expression data analysis. So in this session, we will be doing the analysis using OmicsLogic resources and the Bioinfo platform that we will be talking to you about very soon in the session. So transcriptomics, to give you an overview, is to study organisms' transcriptome. The transcriptome refers as a sum of an organism's RNA transcript, right? So the DNA information that is there in the genome gets converted into RNA through a process called transcription. A segment of DNA that gets transcribed into an RNA molecule is called the transcript unit. So studying and understanding the transcriptome gene expression levels help us understand that what are the proteins or what are the key genes that were maybe expressed differently in a different condition. So it is also about comparing these different expression profiles that we get from different experiments, and that technology and will be also covered, like doing differential gene expression. How would you do that? How can you get from raw data to a structured table of expression first of all, and identify which are the important genes that have shown this variation by using different type of statistical analysis. So we will be learning and we will also learn to use these various statistical methods of data analysis using supervised and unsupervised machine learning that can be applied to biomedical data or different kind of other data types. So using various examples of omics data, we will see how such analysis can yield significant results for various biomedical applications. And I quoted this because it's very important, because when you are looking for information from a very big data, it's like finding needle from the haystack. So significant result is something that is very important and could be, if it is a research, it could be publishable as well. So about the OmicsLogic program, in this OmicsLogic beginners training program, the participants will have an opportunity to learn about various technologies, tools, algorithms, and pipelines, and workflows, and data sets, and research publications. And these are some of the 6 data types that you can see on this slide. So together with this existing modules for which you will gain access for the OmicsLogic Learn portal, where you can explore these modules, exercise the pipeline building. How can you build a pipeline? How can you do coding from the very basic and apply it for bioinformatics, not hardcore programming, and various methods of analysis using project examples from peer-reviewed publication. But to start with this, I mean, we will have to do